order to get market approval for a new drug and its commercial production, a number of necessary phases need to be completed successfully. The manufacture of the active pharmaceutical ingredient and its formulated form need to pass the preclinical phase and then the clinical phases 1 to 3. Thereafter, the new drug application must be filed and finally the API can be supplied commercially upon market approval. Let's go over these phases one by one, step by step, and explain how Bachem can support your successful drug development process. The first step in the development of a new chemical entity is a feasibility study. The target of this study is to evaluate the process basics for the later manufacturing process, such as most suitable starting materials and coupling reagents for the upstream process. For the downstream process, a first evaluation of the cleavage cocktail is performed and related parameters such as anti-solvents for precipitation of the crude peptide, as well as conditions for the purification of the target compound by PrEP-HPLC are evaluated. In this way, it is possible to prove that the desired quality of the material is accessible. In the preclinical phase, some material is required for use in vitro tests and in toxicological studies. Such material is typically produced under non-GMP conditions following the process basis as evaluated in the feasibility study and adapted to the specific challenges of the target compound. It is recommended that the material intended for use in toxicological studies is of relatively low purity, typically between 90 and 95%. This allows to qualify the process-related impurities in respect of toxicological aspects. GMP material suitable for use in phase 1 clinical studies is manufactured following the process as established so far and further adapted based on prior experience and additional knowledge gained in the course of the tox batch manufacturing campaign. It is crucial for this step, as well as for all future process amendments, that the impurity profile of the tox batch is matched, which means no impurity at a higher level than in the tox batch and no new impurity compared to the tox batch. In preparation for phase 2, a process development program is initiated. The goal of this process development is to establish and implement a sustainable and robust manufacturing process suitable for the production of GMP-grade material for use in phase 2 clinical studies. The scope of this process development work package covers the basic strategy of the upstream and the downstream process, such as starting materials including optimization of equivalents required to complete coupling reactions, coupling regions including optimization of volumes required, washing cycles including optimization of volumes required, cleavage cocktail optimization, screening of stationary and mobile phases for PrEP-HPLC purification of the crude peptide, optimization of the isolation conditions for the final API, which means investigating the options lyophilization versus precipitation or crystallization, and last but not at all least for Bachem, to address environmental protection aspects. Every single step of the resulting optimized manufacturing process is described in a developmental master batch production record and is assessed by an FMEA in order to identify potential critical steps of the manufacturing process. Process risks identified in the FMEA, if any, are then eliminated in a second round of process development work packages. Upon completion of the process development, a so-called design batch is produced to prove the suitability of the process for future GMP manufacturing. Then the process is again assessed for potential risks in an FMEA based on the generated process data. The overall outcome of this process development program is the basis for the completion of the master batch production records, which are required for the production of phase 3 material. According to the regulations, material for use in phase 3 clinical studies has to be representative of future materials produced for commercial supply upon market approval. The first step towards preparation for the manufacturing of phase 3 material is to scale up the process to the batch size as agreed with the customer, depending on the estimated needs upon market approval. Then the manufacturing process is characterized at scale by evaluating NORs, so-called normal operating ranges, and PARs, so-called proven acceptable ranges, for all process parameters like, for example, temperatures and volumes. The suitability of the final production process is tested by manufacturing a pre-validation batch. Such material is suitable for use in phase 3 clinical studies. The robustness of the process and the suitability to deliver the target compound in compliance with GMP guidelines will be proven by a process validation, performed at commercial scale on at least 3 independently manufactured product batches. Required process validation plans and reports are prepared based on the final process as developed and established. According to Bochem's approach, the process validation is used to generate additional material required to perform phase 3 clinical studies and for stock building for the go-to-market phase and beyond. The process validation data is summarized in a process validation report, which is the basis for the compilation of the CMC part of the DMF required for an NDA filing. At Bochem, we are committed to supplying drug developers with high-quality API. Therefore, to release any manufactured GMP-grade material according to specifications mutually agreed with our customers, suitable analytical methods are of high importance. Requirements for the analytical methods vary depending on whether the project is in an early or a later clinical phase. We support your NCE project through all development phases, making use of our extensive capabilities relating to the analytical techniques used in the peptide field. For early phase projects, the CMC development concept at Bochem involves the development of a product-specific reverse phase UPLC or HPLC method that should be capable to access the impurity profile of the API. According to our understanding, a well-developed method should allow for determination of related substances from the manufacturing process, and the method should be stability indicating. Further, the method is ideally compatible with mass spectrometry and suitable for assay determination. In preparation to supply GMP material for phase 1, we additionally assess in accordance with pharmacopoeial requirements the suitability of the compendial testing methods for bacterial endotoxins and microbial limits. For most other release methods that are commonly used in peptide analytics and that require validation, we rely on our own generic method validation concept at an early stage of the development. Product-specific validation is then considered at a later stage. This approach allows for an efficient development timeline for our customers. Examples for such methods are the assay test by elemental analysis or testing for residual organic solvents by GC. In later clinical phases, requirements for analytical methods become stricter, and hence the product-specifically developed UPLC or HPLC release method must be validated. Additionally, other release methods, which are thus far only generically validated, should be validated on a product-specific level to verify that for a given product, the methods used are suitable for their intended purpose. For bacterial endotoxins and microbial limits, validation is completed by demonstrating suitability for 3 representative batches of the API. In order to avoid the need for revalidation of any release methods, ideally all methods are validated product-specifically once the manufacturing process is developed and the impurity profile as well as the final drug substance specifications have been established. However, Bochem is committed as well to coordinate analytical activities in a way that can meet drug developers' requirements, such as demanding clinical timelines or a lean development approach. This can, for example, mean to provide material for phase 2 clinical studies before the process development is completed. In such a case, requirements with regards to method development and validation status of the applied methods are tailored towards the release of material for phase 2 clinical studies. Hence, certain methods like assay by elemental analysis, as well as purity and related substances by UPLC, must be validated product-specifically already, while others can be validated product-specifically at a later stage. However, consequentially, a revalidation and sometimes further development of certain already validated methods might become necessary further down the line, once the process, drug substance specifications, and impurity profile have been fully established. Stability studies are conducted throughout the NCE development process in order to provide evidence on how the quality of the drug substance varies with time under the influence of a variety of environmental factors such as temperature and humidity. The collected data can be used to establish a retest period and storage conditions for the API. For early clinical phases, developmental stability studies are typically conducted to get the first indication on the stability of the drug substance or to confirm an assigned retest period or shelf life under potential long-term storage conditions. Such early developmental stability studies can deviate from the ICH guidelines and are conducted in alignment with our customers' requirements. Once the development activities for the drug substance have progressed to the stage of process validation, formal stability studies according to the ICH guidelines are performed with batches produced during process validation. Those studies are used for the purpose of registration, and they are intended to show that the drug substance will remain within specification during the retest period or shelf life if stored under the recommended storage conditions. A representative number of batches, in general 3 batches for new submissions, are stored to generate statistically relevant data on the stability properties of the drug substance. These studies are also conducted at accelerated conditions to determine stability during short-term excursions of the intended storage condition and for additional evidence of the selected retest period. Eventually, the drug product is launched and marketed. This is the time to initiate follow-up stability studies on the drug substance. Those studies are intended to confirm the consistency of produced drug substance batches regarding their stability in the chosen container closure system under defined long-term storage conditions. For commercial products, at least 1 batch per calendar year is foreseen for follow-up stability. The follow-up stability data provides continuous proof of drug substance stability and can also be used to extend the defined retest period. Along with our manufacturing and analytical service, we are committed to support drug developers in finding the best regulatory strategy for their API. Therefore, we not only compile DMFs for commercial applications, but Bachem also writes CMC sections needed for clinical trial applications in close collaboration with our customer, and provides other regulatory support like, for example, documentation review and preparation of responses to authority questions. You are more than welcome to benefit from the expertise we have to offer. Contact us for any questions and inquiries.